Welcome. This is going to be a tutorial on MEG, and so feel free to ask questions along the way. It's meant to be educational and get you up to speed if you haven't seen it before, or if some of it may be a bit lowbrow if you've done MEG, but could be a good refresher. If you have any questions during the talk, please feel free, but if you wouldn't mind either grabbing a mic or I'll repeat your question, whichever you prefer. So the first lesson is actually on the first screen. How many people pronounce those initials or say the initials? MEG. Who's MEG? Who's Team MEG? How about MEG? All right, so that's the first differentiation between our two teams here, red and blue. We'll go with Team MEG and Team MEG. That's actually a bit of a, I wouldn't call it an age test, I would call it a background test. So if you came up in the EEG world, you are used to pronouncing the initials, so you automatically say MEG. So it's awkward to say in a talk EEG and MEG, so you just say MEG. So, but often people came to MEG with no previous EEG experience say MEG. So just something to be aware. You may be grating on the ears of old timers if you say MEG, but it's fine. It's probably better to pronounce it. So this is our system. It's now about 15 years old, still state-of-the-art. There aren't many companies. CTF actually went bankrupt not too long ago and now has been resurrected under a different name. There is a new company called York Industries that is going to be selling machines. I think they've sold some but haven't delivered any yet, and they actually just purchased the NeuroMag system from Electa, so they're actually going to be servicing that. I'm not sure if they're going to be selling it. So it's not a great economic model, MEG, because it's the only clinical application is epilepsy pre-surgical mapping, and there are very few hospitals that have enough volume for it to pay for itself. So then it has to be funded with grants, or you're at a place like NIH that can just purchase it and run it. So it's a unique opportunity to have it. This is a 275 channel system consisting of radial gradiometers. Oriented, as you can see here, radially to the head. There's actually two names for these type of gradiometers. They're actually technically called axial gradiometers, and then the orientation is radial to the scalp. And this is what the inside of this dewar looks like. This is a dewar, essentially a thermos bottle filled with liquid helium to keep all of the wires inside of here superconducting. It's one other reason that this is an expensive system. Currently we have to refill this with helium, and that costs thousands of dollars a week just to keep it filled. We're actually getting a system to repump the helium soon, so that will cut down on costs. So inside each of those cylinders that I showed you are one of these first-order axial gradiometers. But other systems, and in fact ours in its reference sensor setup, has magnetometers as well. So you'll see this in the literature. Different systems consist of different types. The NeuroMag system has magnetometers and planar gradiometers. Ours, in terms of recording from the scalp, has just axial gradiometers. So depending on the paper you read, you might actually be seeing data from different types of setups. These are just very thin wires that pick up, that actually the magnetic field that escapes this very ugly scalp here will generate a microcurrent in this wire, and then that microcurrent will be read out by a squid. This is the kind of the fancy physics thing that actually can read the very tiny currents out without interference, so that the current can keep flowing continuously in those wires, and then that's what we read out into our system. So the source of these signals, at least theoretically, the long apical dendrites of the cortical pyramidal cells are the theoretical source. You obviously can pick up activity from other places. This doesn't have to be the case, but a lot of the initial modeling of source activity was assumed to be from this. These are the things that most look like wires that run in parallel in the head, and then it would be assumed to generate a summed current and then associated magnetic field that you could pick up outside the head. So about 10,000 or so of these cells receiving input at the same time. Again, these are dendrites, so this is input to these layers. was at least previously been estimated to need to generate a source, maybe fewer. And the typical evoked field is about 100 femtotesla, so very small. The comparison to EEG. So we're picking up a result of the exact same activity, theoretically at least, but as you know from physics, every time you have a current flow, you have an associated magnetic field. So if you look at the yellow circle on the left, that would be the magnetic field surrounding a current flow in the dendrite. So if you use the right-hand rule from physics class, my thumb is pointing in the current direction, the magnetic field is surrounding it with my fingers. Sorry. So that magnetic field is what we pick up outside the head. And you can see there are some orientation issues with that. You will best be able to pick up field at a certain orientation if the field is escaping the head, and especially right underneath one of our sensors. EEG is the exact same activity, but actually what you pick up is the extracellular potentials resulting from that current flow. The problem with EEG is that volume conduction distorts these signals before they escape the head, and so it makes source reconstruction much more difficult. You have to have much more accurate modeling of all the different conductive layers that you have in the head, whereas the magnetic fields pass through the skull without interference. There are some aspects of MEG that make EEG better potentially. One is, for instance, the SMA. You have two things wrong with the SMA. Well, actually, probably just one. You actually have a source cancellation effect, where if you have bilateral SMA, the current sources are pointing at each other or away from each other, and the associated magnetic fields wrap in opposite directions and cancel each other out before they even escape the head. There's actually a famous paper where, in our world at least, not famous to everybody, where someone had a stroke and just one SMA, and then they had a beautiful magnetic field from the SMA, but previous to that you couldn't even see it. You do have orientation issues as well. I think previously people were a little strict with their view on this. They said, well, you have to have perfectly tangential sources, so sources oriented perfectly tangentially to the scalp so that the field escapes perfectly, but of course any orientation is going to produce some external field, and especially with full head coverage, you're going to pick it up somewhere. It may just be weaker, and maybe it may be spread out across a lot of channels because of orientation effects, but it's still there.
What's SMA?
SMA is the supplementary motor area. That's what it is. Sorry, I say SMA so much I remember the name. It's bilaterally in premotor cortex, along with central fissure, so that you have the sources pointing at each other. This is an example of MEG. This is actually from our machine. It's we do have an epilepsy group that does pre-surgical mapping, or at least investigatively doing so. And so this is a spike recorded in our system. So this is just raw MEG channels. This is an array of channels, and this is a topograph produced where essentially you flatten out mathematically all the channels, and then you interpolate the signal between the channels, and you end up with a topograph of the signal. Blue in our case is field exiting the head, and red is field entering the head. And so if you look at this, it's a little bit not a perfect orientation, but essentially that means there's activity in between here. So you actually, with our radiogradiometers, peak activity is off to the sides, because remember you have field going in a circle around some source, and that spreads out the further it is away from the sensors. And so you may actually have a positive and negative peak on both sides of a source, and actually directly over the source you get a zero reading. This is a difference between our system and the NeuroMag, where you have planar and radiometers and magnetometers. There, the peak activity is actually directly over the source. So some people who want to look at topographs and just simply work in channel space, the NeuroMag system is a little easier to interpret because of that. At the bottom there actually is a topograph of the EEG, so we can collect simultaneous EEG with our system as well. So you can compare the two, and you can see the source is about where I said. It's in the middle of those two peaks there from the EEG. So if you're conducting time series analysis, which is essentially all that used to be done, before we had whole head coverage and easily available MRIs and so on. What are you looking for? There are several things, but people often look for complex spikes or waveforms. This is an example. It's kind of a facsimile of a signal from a sleep study where you have a K complex that could look like this. And you also have sleep spindles, which are an oscillation, in this case somewhere near the alpha range. So you can also have oscillatory events, some of which you can see in raw data, some that require signal averaging or other sort of processing to pull out. So here's an example of an experiment you might do where you're looking at time series. This is a what's called an evoked or event-related field. Those names are a little bit overlapping, but evoked essentially means what you think it means. It's something associated with a stimulus or a response, and often means things that are phase locked to the stimulus or response, so that you have a perfect timing of an oscillation or a spike after a response. So that means if you do signal averaging, so if you average time series around a response or after a stimulus, you should be able to enhance the evoked activity and reduce anything that's not phase locked or not related to your stimulus. So this is a example of a P300 or an oddball response. And you see the time series is an average time series from one of these channels, probably this one. And you can see this at this time point, 250 milliseconds or so, this is what the topograph looks like. So you can see it starts to getting more difficult to interpret where the sources are, even with a simple experiment like this, the auditory oddball. You have 3 peaks here, and so where exactly is the signal coming from? It becomes a bit of a guessing game unless you do some source localization. Still in the time domain, we can have you can do fancier things. For instance, this is a time frequency analysis of an evoked response to a series of 5 clicks. And you can see time is on the bottom dimension. You have a click every 250 milliseconds. And you can see there is a big response in low frequencies to the first click. And then you have a gamma burst after each individual click in the series. So this is when you have data where just looking at a raw time series, you may have a lot of mixed signal. This is one way to try to pull that apart. So assuming you may want to know where these signals are coming from, how do we do that? So when we record, we actually have 3 head coils that are energized all the time. So we're actually picking up the signal from those and localizing them in space so that we know where the subject is during the run. It's very important that they stay still. This is another disadvantage we have from EEG. The sensors here are not fixed to the subject, so if they move too much, their brain is moving relative to the sensors, and so that distorts your signal. So, but we can tell how much they move during the run, and actually can record continuously where their location is to see when they moved and so on. But those head coils are then used for co-registration. So we take an MRI and we mark those locations on the MRI. It's important if you are ever doing a MEG experiment, you haven't before, that you collect a good structural MRI, not clinical, that has the nose and ears on it so that you can mark these locations. It's often that people clip off these things. The other thing you need is the entire skull. So we actually use that in our modeling. So some people will clip off the top of the skull, for instance, when they collect their slices, and that's bad for us because you have then a incorrect shape for the skull, which is used in our modeling. So how do we localize then once we have these locations? The earliest way to do it, and one that was inherited from EEG, is called a dipole fit. A dipole is essentially a theoretical point current source with a direction. And essentially you do something analogous to a regression. You find the best dipole that fits your data. In this case, it's 2 dipoles. This is the problem with dipole fitting, is you usually have to have a hypothesis ahead of time of how many sources you have. So you have to set the number of dipoles usually when you do this. So this is a nice, fairly easy one where you have an evoked auditory response, and so you know you're going to have activity from both primary auditory cortices, and you can see in the topograph a nice, what we call a dipolar structure is usually what's referred to, because it looks like something you'd easily fit a dipole to, where you have field exiting and entering the head here, which means you have a source in the middle here, which has this orientation, and the same on this side. And you can see this is where the fit turns out fairly accurately in this case. Yep.
Why are they going the opposite on either side?
Well, it's actually because of they were actually oriented the same way, but if you do the right-hand rule on opposite sides, you end up with, in this case, like that, in this case, like that. So you end up with the field entering and exiting the head at opposite points, but it's because of the opposite symmetrical orientation of those sources. Sorry, he asked why they are opposite direction, the dipoles. Sorry for not repeating the question. So that's a simple case, and it works well. It works well if you have a simple kind of evoked type responses where you have a good idea of how many sources you have. But in anything much more complicated, including just the auditory oddball response, where it's not clear how many sources you have, you need something fancier. There are many competing ways to do fancy. Lots of mathematicians and physicists and engineers have come up with different methods. Of course, each person will tell you theirs is the best, but this is the one we use for some reasons. It's not necessarily the best, but I think it is very practical. One practical reason is that the method was invented by the same company that built the machine. So the software came with the machine, although we have our own software now. The other reason is that this method in particular is fairly robust to artifacts. And so here at NIH, where we're dealing a lot of clinical populations, you don't want to lose subjects because of artifacts. So this method is actually fairly good for that. It's called SAM, synthetic aperture magnetometry. No one says SAM, so there's no controversy in pronunciation here. Produces whole brain estimates of source power for a specified time window and frequency band. So here it's not giving you sources at an instant of time, it's over some time window. You actually have to build up enough power in your signal for the method to work. So you need some sort of time window. You also usually specify a frequency band for a couple of reasons. One, there are a lot of different frequencies of activity in the brain. If you try to reconstruct all the sources at all frequencies at once, it's too big a problem for the math. The other is that we know that the brain acts in certain frequency bands ahead of time from other research. So we know, for instance, we might want to be looking for alpha activity in this experiment. So we specify a band and a time window that is experimentally relevant. The method works by creating a spatial filter, we're also called a beamformer, so you may see that in the literature, for each voxel. So it goes through the brain one voxel at a time. You essentially set up a grid of voxels that you set yourself. It's not like fMRI voxels, but it's a similar concept. This is just a grid that you mathematically define. And you go through one voxel at a time, and you set up an individual spatial filter or a beamformer for that particular voxel. So what you do is you estimate what the activity at that voxel would look like at the sensors, and then you develop a spatial filter which does two things. It tries to block out activity from everywhere else in the head, or essentially reduce the power from all the channels, but keep activity from that one spot. And this is of course, a maximization or minimization procedure. You do the best you can, and of course, the better the data is, the better that this procedure will work. If you're trying to there's two ways that this could go wrong. You could you could have not enough data for this math to work, so you have a covariance matrix that is not properly filled out, or you could have too much. You could have too much signal that you're trying to block out, so that the math won't minimize properly. So then once you get this voxel image, you can then perform group statistics in any way that you normally would with fMRI software. We actually use AFNI because they happen to be based here, and they modified their software to read ours, but you can use anything available. So here's an example of a SAM analysis from an NBAC working memory task, and also some resting data for the same subjects. This is the NBAC task. If you're not familiar with it, it's continuous, run in blocks. So you have zero back, where you see one of four numbers, and you respond with the number you just saw. One back, you wait until after the second stimulus, and then you respond with the number you saw one ago, and then two back is the equivalent. Does that make sense? So How do we determine what frequency bands to localize with SAM? You could just do it based on hypotheses from the literature. You can also do some data exploration first. These are power spectra from three of the conditions: a 2-back, 0-back, and rest, to actually analyzing the entire block and getting a power spectra from each block. So this is the amplitude on the left and frequency on the x-axis, and you can see in this case 3 main frequency bands that exhibit task-related activity changes. So you have alpha. You can see at rest you actually have the most alpha, and at 1-back it goes down, and then 2-back, or 0-back, and then 2-back it's the lowest. Beta, the same thing, it's highest at rest. And then you have this band, what we're referring to as high gamma. It has other names, where rest is actually the lowest, and this is a zoomed in version of that. You have rest, and then 0-back, and then 2-back at these very high frequencies. These are frequencies that haven't been typically analyzed with MEG, one because of data, the size of the data set you have to record at high frequencies, but also just because the signal to noise there is not good, so it's often assumed you can't really see much there. So this is an example of something I'll describe again, but I'll describe it right now. Why not? So you have in the frequency space in the brain, you have you can have frequencies that go up or down, relatively speaking, depending on a task. And they are often called either synchronization or desynchronization. Those are somewhat loaded terms because they imply that you have an oscillation that's ongoing that either synchronizes, would mean that you have a patch of cortex that all starts oscillating together during a task. Desynchronization means you might have a patch of cortex where the oscillations break up and start canceling each other out. That's not necessarily what's going on. These terms are now used to generally mean increased or decreased oscillations. It may not necessarily mean that theoretical construct that I just described. But if you look at this, the alpha activity, you can see 2-back greater than 0-back. So you actually see blue, meaning you have activity that goes down in the 2-back. So you actually have what's referred to as desynchronization, which is a common phenomenon in the brain. You actually have, it's very well known that you have visual and motor desynchronization after either a visual stimuli or a motor movement. So these are well known to exist, and they mean increased activity or activation in that area. You can also have increases in the red, that areas that increase with the task. The high gamma, you can see it's in slightly different areas, which is interesting. And most of the activity is actually an increase, as opposed to a lot of it is decrease here. There's some idea, at least, that high gamma is not really an oscillation in a traditional sense, so synchronization and desynchronization wouldn't be accurate terms to use. It may possibly be the aggregate of spiking activity in the cortex. It could be dendritic summation, essentially, of all the spiking coming in. And that's why you end up with a flat spectrum, as you see here in the high gamma range, as opposed to a peak at a certain frequency like alpha and beta. So it's an example of the SAM analysis. Any questions on that? So how does that analysis compare to MEG? Here's an example showing that, in fact, desynchronization or a reduction in activity can actually correspond to increased bold. This is the same 12 subjects with the n-back task. It's 2-back versus 0-back again. And you can see here it's a slightly different analysis. It's a 500 millisecond window centered on the responses during the task. And you get a desynchronization pattern on the left in MEG that is relatively similar to the increase in bold on the right for the same subjects. You can see the localization is not quite as good. We're not claiming with MEG that we can localize things in better than the bold signal for most cases. But we have gained some information here. We know that, at least in this case, for these areas, the bold signal is corresponding to a decrease in beta activity. So that's one piece of information we picked up. This is only in this case. Other experiments have shown other types of activity corresponding with bold. You can also get simply evoked activity that corresponds with bold, not a frequency-based activation. So this is still a lot of work to be done to really determine what directly relates to the bold signal in terms of oscillations, or maybe it's just everything. Here's an example of, though, what Why would you do MEG if it's just slightly worse source localization than fMRI? One reason, of course, is that we have better temporal resolution in most cases. So this is actually an example of doing the SAM analysis in a sliding window fashion. So you actually take your time window and you run the analysis independently over time windows that slide forward in time. And so you can actually get a smooth representation of the brain activity over time. So it's actually from a tone duration experiment where a subject, in the case on the upper left time frequency plot, they were supposed to respond if they heard a tone that was 0.6 seconds long. And you can see that big blue burst is actually the beta and alpha desynchronization that you get with movement. So this is a movement signal. We know they press the button. Or they were supposed to not respond if it was a 1.2 second long tone. In this case, there is no motor response, but you still do see some low frequency activity and also a beta band activity here. So it's alpha and beta. But this is a beta analysis. So this is actually looking at beta band with a sliding window for the tone, tone that doesn't require a response. You actually see in these different windows the activation developing, peaking in amplitude at least somewhere around when the tone may have shut off, and they may have been required to respond. They actually don't need to respond, so you can see the activity going away even before the end of the tone, which happens at 1200 milliseconds. And that was the same video I showed at the beginning of the talk. This is actually taking the same data and playing it in a loop, so you can see the activity. So this is about 2 seconds of data. You can tell essentially the video is on a loop on a GIF, but when you see some yellow activity in the auditory cortex right there, that's when the tone comes on. That's the auditory evoked response. The rest of this then is them listening to the tone and determining whether or not they have to move. And you can see motor cortex activity even though they aren't actually moving in this case, maybe suppressing a movement. So what else can we see with MEG? There's been in literature, I think, based on old technology, I've actually seen it. We had a IRTA show up once that had brought his neuroscience textbook in, and it had a paragraph on MEG, and it said you can't see deep sources with MEG, period, no qualifications, which was incorrect. But this was based on old technology where you had very few channels, and also the technology has developed where the signal to noise is much better. We also have reference channels now that block out a lot of environmental noise. So you have a much better signal now. And so it's really just a matter of signal to noise. Obviously the magnetic field from a deeper source is weaker by the time it escapes the head. So it's harder to see, but you can see it with appropriate experiments and analysis. Here's some examples from several. The one on the left hasn't been published yet, but the other two have been published. The amygdala and the hippocampus activity were in experiments, as I'll show, that were known to elicit activity in those regions, and so it was, and at frequency bands that were known as well. So it's fairly reliable that we're seeing those locations. Connectivity. This, of course, has been a hot topic and can be done with MEG. There's been papers published with it. The problem in the literature is that there isn't really an established method or really a theory behind what your method means. So what exactly is connectivity MEG? It could result from a lot of different things. This is actually from the NBAC again. It's power envelope correlation. So you actually take, similar to that SAM analysis where you take a sliding window, so you actually take the envelope of power. So how does beta change on a slower fashion over a sliding window of time? And you take that as a time course at every voxel, and then you correlate those time courses between voxels. So you actually see in this case, it's essentially what areas are active at the same time in a kind of a smooth sense. And then this is coherence with the exact same data. So this is actually, so you can see, depending on the method, you can get a very different pattern. So it's going to be, it's kind of buyer beware with this. And, you need to really make sure you know what your method means in terms of your data, and then be aware that if you tried another method, you might get something else. So coherence is actually, if you take the actual estimated signal. So you use SAM to estimate a virtual channel, so an estimated signal at the source, and then you see if the oscillations in two different regions are oscillating in phase with each other. So they're actually coherently oscillating across regions. So that's the end of kind of the intro to MEG, and I'm going to go through some examples. Any questions so far? So here, at least, we're doing clinical research, and so what are people typically looking for? As I've shown you some examples of the type of responses we can see.
Sorry, can I add
Yep.
about the previous connectivity?
Yep.
So have people in MEG been able to replicate like the most prominent networks, like default mode, salience, and so on? Sorry if you covered this.
Yeah, so he's asking if people have been able to replicate default mode and other networks. Yeah, I believe that people have seen similar networks, and I don't recall offhand which method they would use. But yeah, that has been done. It's you know, and there may be a lot of things. You know, coherence and power envelope correlation could show the exact same areas. They see some pairs across the top frontal cortex. So it may not be, you know, the method just, it may depend on the sensitivity. You could be seeing the same underlying activity. It's just whether or not you're able to make out a lot of the pieces of the network. There's always that the pattern of the problem, and it's probably true with fMRI as well, that you could have a false positive caused by coincident activation, caused by innervation from a third source, and especially in MEG where it's relatively difficult to see thalamus. So you could have thalamus input to, say, both auditory cortices. We know that's coming from thalamus. They're not connected to each other at the time. It's an early evoked response. But if you did a coherence, they would be perfectly correlated with each other, and you'd say, connectivity, but it's really false. So it's always a bit difficult to interpret, but still possible. You're also, again, relying on virtual channels in this case. We don't actually have electrodes in the head. And so we're estimating thousands of channels based on 275 real channels outside the head. So you're always going to have, even in your estimate, some correlation with neighboring areas and so on. That's just based on your source localization. So it's always difficult to do connectivity between neighboring areas because it's probably the same source that's just on all the different channels, that sort of thing. But just problems to be aware of, but it's certainly possible. So again, that goes into what. Did that answer your question? Okay.
Yes. Okay.
This goes into, you know, what can we look for? Again, we can have delayed or reduced amplitude evoked responses. This is even in EEG in the early days. We often look in patient populations and say, hey, their evoked auditory response is reduced or so on. You could have oscillatory power changes, some of the beta or gamma range that we saw. You can have, as we say, long-range communication differences, connectivity, and also simply conduction delays that may cause delay in evoked responses. Some of the early so the clinical work with EEG is looking at delays in brainstem potentials that may be caused by problems caused by strokes or different things. So there is simply delay measures that you can use as well as connectivity in some of these things. You can also do more complex measures. We have a researcher using entropy to try to look at look for signatures of differences in disease. So here's an example of differences you might see in patient populations from our lab in the schizophrenia branch. This is at rest, seeing something close to the default mode network areas, I guess. You have a difference in the gamma range, so actually less gamma in patients at rest in this area. So if we know we can see differences in patient populations, what can we then associate these differences with? Of course, many of these things you can do with other technologies, but these are some of the things that have been attempted in our lab. There's some that, of course, you could do with a variety of technologies. Some may be specific to EEG and MEG network type models, and neurotransmitter levels are an interesting one. There are some people have modeled where beta and gamma activity come from, for instance, and then you could relate the frequency and amplitude of those oscillations to neurotransmitter levels. For instance, GABA may affect gamma frequency and amplitude, and that might be related to disease. So that's an example of one thing that could be where you could associate imaging, MRS for instance, and mathematical modeling and MEG in an experiment. And I'll show you a few examples of things that have been done. This is an early example of using genotype COMT that could be involved, that is involved in dopamine signaling, and we can see different genotypes. We have a different amount of beta desynchronization during the 2-back versus 0-back again, so that's similar pattern. This is an example of from a depression study, the one where we saw some hippocampal activity during this is a spatial navigation task, and we see differences between the patient normals and patients. And you also see, even within the different groups, you can see on the right, that's a regression with the amount of theta power. It's a low frequency, 48 hertz, in hippocampus. The amount relates to how long it took them to find a hidden platform in this spatial navigation task. And you can see that related with a slightly different slope in both groups. Here's an example, hasn't been being written up now, but this is back to that P300 or the auditory oddball response. You can actually do, this is a case again of a sliding window SAM where you try to recover some temporal information. And we can look at differences here between patients with schizophrenia and healthy controls, and we see a latency and amplitude difference in several areas. This is a result of an ANOVA where you see an interaction of amplitude and latency at some of these spots. So this location, you can see patients have a later and lower amplitude oddball response. And this is another location. And we went on further from that and used the same data to see if there's any correlation with different factors from neuropsych testing and symptoms. You can see there's in cerebellum we have a negative correlation with general intelligence, and in this insula ACC kind of area we have the suspiciousness scale on the PANS correlates positively. Here's another example of this is a pharmaceutical response in MEG. This is the ketamine experiment, and they're looking at amygdala and other areas, anterior cingulate, that could predict the ketamine response as an antidepressant in patients. And you can see an actual, on the left-hand graph, that's on the y-axis, that's the change in their depression score, and that's beta power on the right. So you can see a positive correlation in the ACC with whether they respond to the ketamine and the beta power in that area during a working memory task. And then on the right is actually a bit of a connectivity result. It's actually the coherence between ACC and left amygdala. And you can see that coherence measure is negatively correlated with that depression response. Here's another interesting result from a different lab. This is using TDCS, direct current stimulation during MEG, which is pretty interesting that you can do this. So they actually do sham or active direct current stimulation. You can see the locations at the top, and they're doing visual stimulation at the same time. And you can see that they have differences in both I believe is the beta and alpha and gamma range, I believe, during the, is it just alpha, yeah, gamma and alpha range during visual stimulation. So if you do the direct current stimulation, you can actually produce an offset in the amplitude of the gamma and the alpha activity after visual stimulation, which is pretty interesting. And then the last one, this is one that was done a while ago in our lab. This is actually a brain-computer interface experiment where the subject was a stroke patient actually learning to control a prosthesis with their brain. So they didn't have control of their arm, and so they actually had a robotic arm or over their real arm that would open and close their hands, and they were trying to learn how to do that with their brain, and then actually recording what areas were active in the brain while they were trying to do that, to try to pick up what activity they were using to train the device. And it turned out it was alpha activity mostly in frontal cortex. So you can see they thought maybe different parts of motor cortex might be reorganized to do this, but they actually were using frontal cortical alpha activity to control the device. And so the future, this is the last slide, but some new engineering has occurred where EEG could possibly become more common and much cheaper because it wouldn't require liquid helium. So you could actually do room temperature MEG. These are optically pumped magnetometers at room temperature that actually use a laser and spin polarized rubidium atoms in a vapor, and then you actually pick up the magnetic field that way. And we actually have some of these sensors, and we're testing them now. It's still in its early phase. We have it's only a few channel system right now, so it's similar to the early days of MEG back in the 80s, early 90s, where you had a few channels and the data is not terrific, but has potential, and it could make these also could be more portable. It could be more like EEG. You could actually attach them to a helmet that would be placed on the person's head, and they could walk around with it, so it could be much more practical than our current system. But it's still in development. That's it. That's my email. That's our website with lots more information. And we have a MEG Announce listserv if you'd like to get updates on MEG. And then also at the bottom, we're going to be this fall starting something related to a journal club, a little more broad than that, and that will be on the MEG Announce list if you want to sign up for it. It'll be more of a sharing and learning opportunity where people can share their, say, experimental ideas or their preliminary results, or if it's a journal article that you're interested in and want to do the traditional journal club, that would be fine as well, but hopefully a forum where we all can kind of learn from each other and trade ideas. So hopefully you'll show up to that. Thank you. Any questions? No problem if there aren't any. It's Friday.
In terms of how comfortable it is to sit in MEG for a long time, is it better or worse than MRI?
Well, so we can actually do seated and lying down. So that depends. The MEG is relatively comfortable. You're seated. You're not inside a closed space. You have to keep your head still, and you're in a helmet. But we don't have the claustrophobia problems, I would say, MRI does. I mean, it's the same. It's not the best, most comfortable chair in the world. It's a similar, like, thin padding as an MRI. But I would say really the claustrophobia is the main difference. So we have very few refusals even from patients in terms of getting a run. Lying down is tough because you're on a bed, but your head is on the hard plastic of the helmet, so it's not the most comfortable position you would think it would be. They do that in epilepsy only because they want them slightly drowsy to get good spiking activity. But the other problem is that the runs, of course, MRI you have movement as well, but you can correct for movement a little easier with fMRI. So usually you want your experiment max around 10 minutes between onset and offset of recording, and then you might want another run, but they may want to move around a little bit. It's just hard to keep still that long, so that is a bit of a limiting factor. EEG, you could run for hours. They could move around all they want. That's one advantage it has. Anything else? If not, thank you very much. Happy Friday.